This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode number 300.
Woo! Congratulations, Vincent.
Well, congratulations, everybody. Recorded on December 14th, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from South Carolina, Michael Schmidt.
Hello, everyone.
From Michigan, Michele Swanson.
Hello. Happy to be here.
And from Missouri, Petra Levin.
Happy to be here.
For number 300. We have a ways to go before 1,000, but 300 is a good one. We seems like we did 200 not too long ago.
Yeah. I just was looking at your notes that I had first joined on episode 56.
Yep.
And I'm amazed that we've already done another 250 just about. Wow. Seems like only yesterday.
Mm-hmm.
I know. I think it's a testimony to people being interested in science that we're still going. And look at all the excitement we had at the last episode in Phoenix at ASMQ. That was great, right?
It was great.
That was
really
fun.
neat to interact with people who listen to our podcast.
And who use it. Yeah, who use it in their teaching.
They use it.
Right.
I think that's fantastic. Yeah.
NSF-funded research.
Yeah. It was great. It was a good time. It was worth going to. Well, this is number 300. We have a surprise for you in a bit. But meanwhile, we began TWiM on February 23rd, 2011. The hosts were me, Cliff Mintz, Michael Schmidt, and Stan Malloy. We've had changes over the years.
Originally, Ron Atlas was going to be one of our co-hosts, but Ron never made it on the air with us. He was very busy at the time, and he never found the time to join us.
Yeah, that's right. He was never on TWiM?
Never. I guilted him at one poster session at the microbe meeting. I said, "When are you going to come on?"
He's right. He's not been on. I'm surprised.
Yeah.
Anyway, so 2011, how many years ago is that? 9. I'm not very
Over
good at
10.
that. Over 10
Over
years.
10.
We'll call it a dozen.
We'll call it a dozen. All right, we're going to start with a snippet from Michael, which, in a way echoes our last episode, right, Michael?
This is a real treat. The title of the paper is Antimicrobial Activity of Panarthrobacterium, or Arthrobacter nicotivorans. And it's from the Northeast Wisconsin Technical College, which is located in Green Bay, Wisconsin. And the authors are Justina Kakel, Maynor Vang, Drew Swanson, Tracy Steno, and Angelo Kolikitas. And it will come as no surprise to our regular listeners that antibiotic resistance is one of the biggest challenges facing medicine today. The whys and how behind the emergence of this antibiotic resistance as a global crisis can be summed up pretty directly in that when exposed to antimicrobials, the microbes either need to adapt or die. And if we have not stressed enough over the last 300 episodes of this podcast, is that microbes are resilient. And with apologies to Michael Crichton, who offered in Jurassic Park, nature or microbes will always find a way. However, what we have not covered to the same extent that it has been talked about and advocated for on TWiV is how we all have a responsibility to encourage policymakers and funders in the supporting the discovery, development, and evaluation of new antimicrobial molecules that could help alleviate what many are forecasting could have as dire of consequence to the human race as global climate change. And that is, of course, the emergence of antibiotic resistance. Well, with that as a prelude, let me welcome you all again to today's paper, which comes from us from the Northeast Wisconsin Technical College that's in Green Bay. This paper is a consequence of the Tiny Earth program, which is a program whose intent is to inspire students to engage in scientific research while addressing some of the most pressing global challenges of our century, namely the diminishing supply of effective antibiotics, the topic that this paper will cover, a rapid decline in soil health, namely how the human race is going to continue to feed ourselves, and an underrepresentation of diversity among scientists, something that we all need to be actively pursuing and encouraging any opportunity we get to encourage all folks interested in science to join our ranks. Tiny Earth's low-cost curriculum immerses students in the thrill of research, and we certainly saw that by talking to our colleagues at ASMQ. Many of those individuals at Q are in the Tiny Earth program, and Tiny Earth supports this process by instructor training. This is a program that has empowered more than 700 instructors to teach at its diverse institutions, ranging from institutions like Northeast Wisconsin Technical College and colleges and even research universities. Enter our research team. Here, Justina Kakel and Maynard Vang set out to learn if there were any antibiotic-producing microbes in a 6.4-acre public park located within the limits of Green Bay, Wisconsin. This park happens to be the largest developed park in the Brown County Park System, where it is described as a quiet serenity with gently rolling hills and wooded areas enhanced by the slow-flowing Duck Creek. They even have an 18-hole disc golf course and a 10,000 square foot formal garden setup. And from the images I've seen on the internet, it's quite lovely. And how I discovered this paper is my nephew, who happens to live in Green Bay, sent me the news article that the local paper did on these two young investigators and their project. It was really a very lovely article highlighting the two young scientists and their mentor and what they were able to accomplish. So I'm going to share with you their paper that made it through peer review and was published in September. Yes, please.
Before you do, that beautiful park that you described, the land was donated by the Pamperum family. And as it happens, the first author of our paper, her fiance's grandmother is actually a relative of the Pamperums.
I don't know if that would be classified under NIH's conflict of interest guidelines.
No, I mean, it's just how happy must that family be to know that
the thing their ancestors did those generations ago, donate or gifting that 6.4 acres to the city, has actually may pay a dividend for mankind. I mean, besides the beauty and the serenity that it has offered to the community already.
Yeah. So tell us more. What is in the soil there?
So here our investigators simply collected soil, whereupon they screened and isolated microbes, something that's done across our country. I remember as a TA in the biology of prokaryotes, sending students out to the fields of Indiana to collect soil and actually see what they could uncover. Well, these students asked the simple question, could they isolate microbes and produce antimicrobials, where their stated goal was to contribute to the discovery of antibiotic-producing microbes in hopes of addressing the global crisis of antibiotic resistance. Well, our investigators isolated, in honor of our special guest who will be here in a moment, the bottom line, the investigators isolated one such microbe, the Panarthrobacter nicotivorans, which is a gram-positive aerobic bacterium that has the ability to degrade atrazine, nicotine, and creatine. And it was originally isolated in North Korea. And that's the news article, not the peer-reviewed paper that offered us, but the newspaper story. So even local papers still are giving us really neat things. And nicotine, I learned, is quite common in our soils. I didn't know that soils smoked, but apparently nicotine is quite common in
What's the
soil.
source of nicotine in soil?
Yeah.
That I didn't dig out, so I don't know, but that was in the news article.
So how did they find this
Their
wondrous
approach,
microbe?
their approach was elegant in simplicity and in utility, and they were able to screen for antibiotic production by simply patching 20 bacterial colonies that they isolated from soil onto a luria broth plate. Many undergraduate laboratories have a plethora of luria broth plates, and upon patching them, they then transferred them onto another LB plate that had a lawn of Erinia kerativora, where they simply looked for an absence of the lawn approaching their isolate. This was easily visualized, and it can be seen in figure 1 of their open source paper, namely panel B.
And this was by incubating the two together for a day.
Yes, for 24 hours.
To give the time for these soil isolates to grow and produce whatever they may be producing, and then see what impact that has on the attempt by the lawn to grow.
And they're just simply asking a question. Do they see any zone of inhibition? And they then took the isolate that gave them this encouraging observation when it was challenged by Erwinia, and then they patched it on top of escape pathogen-safe relatives. Now recall that escape is an acronym comprising the scientific names of 6 highly virulent and antibiotic-resistant bacterial pathogens that include Enterococcus faecium, a common gram-positive organism that many of us appreciate causes that evil antibiotic-resistant infection in hospitals, Staph aureus, Klebsiella pneumonia, Acinetobacter baumannii, Pseudomonas aeruginosa, Enterobacter species, as well as Escherichia coli.
You know what I learned? That E. coli is actually, in a Lancet article, E. coli is actually the biggest cause of death associated with or directly due to antibiotic resistant pathogens.
Yes.
I know. Nobody who talks about E. coli ever mentions that. But
well, we tell our students it's safe.
Well, the ones we work on are safe.
Yes.
But I thought that was really interesting, probably mostly like Klebsiella, and actually most of those are hospital acquired for the most part problems. But I think E. coli should get a little more, I guess, not love, the opposite of love.
And we had a TWiM on this very topic. This was an episode we recorded at the general meeting in which we had an infectious disease physician talking about these antibiotic-resistant E. coli that were causing urinary tract infections, and how a sister was visiting her ill sister who was in the hospital with this UTI, and the sister acquired the UTI because they shared the common bathroom that the ill sister was using, and the sister that caught the antibiotic-resistant E. coli from just simply using the same restroom ended up passing away.
Oh no!
These things are not to be trifled with. Once E. coli gets those sets of instructions, it's a pretty mean and nasty pathogen.
You mean instructions to
resist antimicrobials?
Yeah.
So these investigators rightly screened their compounds or their isolates against this group of gram-positive and gram-negative bacteria, as they are appreciated. These escape pathogens are appreciated for their abilities to evade or escape. So their central hypothesis is, let's look for a drug that will be active against agents for which we need new drugs. And so, the escape pathogens are known for their ability to be multi-drug resistant.
I thought it was really clever, too, that they, of course, didn't distribute to these undergraduates the actual escape virulent human pathogens, but rather close relatives that are known to be safe, but yet will have many of the same attributes.
And the organisms they used is Staph epidermidis, which is a common skin isolate, Pseudomonas putida, which is a garden variety Pseudomonas that you can easily isolate from soil, Enterobacter aerogenes, probably the tame one. One that I found interesting, they also looked for mycobacterial compounds against mycobacteria. They use Mycobacterium smegmatis because, as we know, we're having totally drug-resistant Mycobacterium tuberculosis. Bacillus subtilis, which is a common gram-positive, and Acinetobacter bacilli, which is not Bomani, it's just Acinetobacter, and it's probably a tamer version. The Erwinia that they used in their original screen, and of course, probably a laboratory variant of Escherichia. So again, they were looking for clearing, and they found it. Bacteria in spots 2 and 17 showed some clearing, but they settled on spot 2. And that was after 24 hours, and they hereafter referred to their spot in their paper, if you're following along, as MV2, and it was then ultimately selected for further analysis. They learned their identity of their isolate through a technique that we've talked often on TWiM about, and that was principally they isolated the colony, they isolated total nucleic acid from the bacterium, and then they used PCR primers against the 16S ribosomal RNA gene in order to identify the microbe. Again, recall that the 16S ribosomal RNA gene is ubiquitous in the prokaryotic kingdom, and they determined from that analysis after sequencing the closest relative to MV2 was this isolate, Panarthrobacter nicotivorians, and they found through using an NIH resource, namely BLAST, that it was 99.73% identical to the reference strain.
At that locus.
At the 16S ribosomal RNA gene. Mm-hmm. Correct, Michele. And then they did the next step, and this is important. They assessed its phenotypic traits, where they learned it was indeed gram-positive, and the bacterium showed starch hydrolysis, which is a hallmark of Panarthrobacter, and it was also oxidase positive. And that corresponded by the phenotypic characteristics for the type species that has been deposited. Next, they went down the rabbit hole of doing chemistry. They next isolated the secondary metabolites by inoculating the isolate, MV2, onto an ALB plate. They allowed it to grow, and then their methods are very detailed and straightforward, and they're simple. They tell you how they extracted the colonies, and it's elegant in its simplicity. They simply put their agar plate into a -80 freezer and froze the sucker. Mm-hmm. They then cut it up, and then they did their extraction with the organic solvents, and they were able to isolate an antibiotic fraction. And they had a couple of fractions that they then tested, again, using the same technique of growing a lawn and then spotting their antimicrobial fractions onto it. And they go through details about how they reconstituted them in methanol at a concentration of 10 micrograms per milliliter. And what they did is they spotted 10 microliters onto the agar surfaces, which is approximately 100 micrograms of whatever's in that magic supernatant, because they didn't purify the particular compounds. They then contrasted the results they saw on the Petri plates with their compounds against two controls. One, they used an antibiotic that inhibits peptidoglycan synthesis, penicillin, to look for activity against gram-positives. And then they used an antibiotic, namely tobramycin, which targets the construction of the 70S ribosome by inhibiting the 30 and 50S from coming together. And Tobra will prevent that. And their activity of their 100 micrograms that they spotted onto their plates was equivalent in activity to the positive controls of the penicillin and the tobramycin that they use. And this is beautifully shown on their color figure. And you can actually see the remarkable demonstration that they did indeed have activity. They conclude this paper by offering the value that Tiny Earth afforded them as young investigators. And then they did something that is really, I think, so important. They published the results. They wrote it up and they literally threw it over the transom to the journal. And they asked the question, will you review it and will you publish it? And the answer was in the affirmative, because that is the paper that we are offering for you. And again, they went on to say in their discussion that this Paniarthrobacterium nicotivorans has been described previously for its potential for antibiotic development because it was only tested for the production of silver nanoparticles. And silver, as many of you know, is a strong antimicrobial agent. It's used in many burn dressings and nanoparticles. So the silver precipitates simply because the bacterium is likely donating electrons to the silver ions that the microbe is associated with, and it then precipitates out as elemental silver particles. And this is a common behavior of how bacteria routinely detoxify themselves against toxic metals. They concluded their paper by offering that, to their knowledge, no other members of Paniarthrobacteria's genus have been determined to produce antibiotics. So this is a first for these offers. No one's described the ability to produce these antibiotics. So here, through their efforts, they have isolated a bacterium that may represent a novel source of a new antimicrobial substance that had activity against several pathogens. And so this park, not only did the relatives many generations ago give it to Green Bay, but now the park is giving to the human race by the potential of giving us a clue as to a bacterium that may have the potential to generate a new antimicrobial. They sent the strain down to the University of Wisconsin at Madison for further characterization, and here's hoping that they actually work it up and that they find that it is indeed a good candidate for drug development.
What I love about this Tiny Earth project is there were students that maybe you learn about classic microbiology techniques, plating, looking at inhibition, and then standard genetics, molecular genetics, the 16S, and then getting into a little computational biology, and then separately the biochemistry to extract compounds and see if they had activity. And so this was a team where some maybe were taking a microbiology course and participated. Other students were taking a chemistry class, others perhaps a microbial or a molecular genetics class, and they collaborated to produce this complete story. It's really great.
It is really cool. I also think it's cool because I think we haven't described what, especially for people who don't live in the United States, what Green Bay's climate is. It's not an exotic, tropical location, right? It's very, very cold much of the year.
It freezes.
It freezes very solid. And this is
Well,
if you've ever seen the movie Grumpy Old Men, where Jack Lemmon and Walter Matthau are ice fishermen out on the frozen lake, that is a common behavior in northern Minnesota and Green Bay. When the Green Bay freezes, they build these ice houses. So Green Bay, which is a large body of water, freezes, and they have ice houses on.
Yeah, it's actually a bay on Lake Michigan, and and in Wisconsin, and there's a football team there called the Packers, and their field is sometimes called the frozen tundra of Lambeau Field.
And that field, Lambeau Field, is actually the site of the 2023 Winter Symposium for Tiny Earth. They had their in-person conference at Lambeau Field on December 11th, so just a couple days ago. And actually today, they're having another virtual symposium that you can log on to via Zoom. Unfortunately, it's going to be over by the time we finish recording this episode. But how great that the students are not only getting to do discovery and learn some fundamental concept in science, but also share their work in a peer-reviewed paper, the popular press, and then also present it at a meeting and meet other students who are similarly engaged.
No, it's great.
I think we need to give a shout out to Joe Handelson. Anybody want to talk about the birth of this Tiny Earth program?
It's literally one of Joe's offspring.
Yeah, so she is the Villas Research Professor of Plant Pathology at Wisconsin-Madison. She was formerly the Associate Director for Science at the White House Office of Science and Technology. And she conceived of this kind of active learning project that has now got funding and is being carried out worldwide. So according to their website, more than 700 instructors are engaged. More than 80 are trained each year. And they have engaged 14,000 students per year from 500 different institutions to do this citizen science, this discovery work, which is what we need, right? We need to search broadly in the world of microbes to come up with some new candidate antibiotics. So hats off to Jo for her leadership as a teacher and also policy maker. And she's a former president of ASM as well.
It was so cool. And none of the reagents involved here are very expensive.
Yeah.
I mean, LB plates are about a quarter. The ethyl acetate is extremely inexpensive. They only use 15 milliliters to isolate the material off of the plates. And methanol is relatively inexpensive as well. So again, this is not expensive science to conduct, and you can really, and everybody's soil is different. So if you think, well, they found it, so what's left for me to do? Don't look at it that way. Everybody's soil is a bit different.
I think the Phage Hunters project has been going for, 15, 20 years, where they pick, and I don't think they've ever come up with the identical phage. And
tell us more about that, Petra. Tell us more about that citizen science project.
So it's Phage Hunters is project that's actually started by Graham Hatfull, and we've talked about it when we've talked about phage therapy. It's HHMI funded now. Originally it wasn't. Sometimes it's called C-phages, and it's in place at a lot of undergraduate institutions in the United States and abroad. And the students at WashU, it's a freshman class, but they go out and they isolate from the soil bacterial viruses, phage, that target used to be mycobacteria. I think now we're doing streptomyces. I think actually on the last TWiM episode, they talked about a WashU, one that was isolated at WashU, the vampire phage, but our C-phages students. And the bacterial phage, the students isolate a phage that targets one of these, and they characterize it and they sequence it and they take pictures of them on the microscope and they name them. And some of these phages have been used by Graham Hatfull and colleagues for some of their therapy, their phage therapy against Mycobacteria abscessus, which can be a problem in immunocompromised patients, especially transplant patients. And that's a really fantastic course, and it's had a lot of the students we have who become interested in microbiology as first-year students at WashU come from the C-phages course that we have, or we call it Phage Hunters. It's another great citizen science.
And I think it demystifies the microbiology when you have inquiry-based education. You learn that microbiology is not complicated names. It's really pretty cool stuff that you don't necessarily need to memorize your way through. You can really begin to understand the the beauty of biology as you go through this.
Right. And recognize its value.
Yes,
You're not
absolutely.
learning these just for learning's sake, but they're actually applications that are really important.
Right. And I think both this and the Tiny Earth Project, one of the things I like about it and what I like about undergraduate research in general is even if, any result, and what I loved as an undergrad, any result that you get in labs is often the first time somebody has seen that. Even if it's a mistake, sometimes it's the first time somebody's made that particular mistake. And so like for these students in Green Bay or for students in the phage hunters, like when they isolate that bacterial virus or they isolate a bacteria that produces an antibiotic, it's unlikely to be identical to something somebody did before. So they are the first people seeing that and finding that. And that's a really, I think, fun, exciting, but also a powerful feeling that you yourself, even at an early stage, very early stage in your career, can actually find something new and make a difference. And it just highlights how much there is to do in biology and how we haven't solved anything and how much more there is to learn. So
big thanks to all our colleagues out there that are volunteering as instructors for these two citizen science projects. It's great.
Now we just wait for Elio.
Hi, Elio. How are you?
Hi, Petra.
You joined us on episode number 10.
I did.
And you brought along Margaret McFall-Nye, and that was the episode that we talked about her bobtail squids and how they were able to cloak themselves using Vibrio fischeri.
That's right. Yeah.
And then your charm managed to convince Margaret to join our team.
How about that? I'm very good at giving other people something to do.
A true leader.
True. Here we are.
Yes.
So my memories are very fond, but they're vague. The problem. It's called old age.
Yes.
I'm only 95 years old. 96 years old.
Good for you. Well, Elio, we loved your time here, so we thought it was great to have you on for 300 because it's quite a milestone. So thanks for joining us.
Yeah, we wanted to take a moment to honor our late colleague, Barbara Iglewski, who passed away on December 10th. Dr. Barbara Hotham Iglewski, was born in Pennsylvania, and she was the daughter of a country doctor who had a microscope in his office. And so as a child, she had opportunities to begin to explore the wonders of life. She earned a bachelor's in biology from Allegheny College and then went to Penn State for her graduate degree. As an undergraduate, she was at Allegheny College, also in Pennsylvania. And she earned a PhD there and also met and married a fellow graduate student. They then moved together to Oregon Health Sciences University in Portland. He got a job as an assistant professor, and they brought her on as an instructor at a salary that was 40% lower than his. And she will remember that later in her career. So in 1986, she was hired as the chair of microbiology and immunology at the University of Rochester School of Medicine and Dentistry, and she led that department for more than 23 years. And one of the many successful initiatives was to establish equitable salaries for their colleagues, regardless of gender. She also served as the vice provost for research and graduate education for 3 years, and was the first female at University of Rochester School of Medicine to serve either as a department chair or as the vice provost. So this is just one example of the barriers that she broke. She was elected a fellow of the American Academy of Microbiology in 1986. In 1987, she served as the president of our society. And then in 1990, she took on the chair of the ASM Publications Board, a position she held for 9 years. And her activism continued. She wanted to make a point of getting women appointed as editors-in-chief and also to editorial boards, so there was more equitable representation in leadership roles. She had a long and illustrious career, and I'll describe that research in a minute, but in 1999, she won the Arthur Kornberg Research Award. In 2001, the Susan B. Anthony Lifetime Achievement Award. In 2009, the Lifetime Mentoring Award from the University of Rochester. And in 2015, was elected to the National Women's Hall of Fame, which is based in Seneca Falls, the birthplace of the women's movement. So really great. So her research, she started out studying diphtheria toxin, published on that in the 70s, but really then turned her attention to Pseudomonas aeruginosa. So this is a versatile environmental bacterial pathogen that causes a wide range of opportunistic infections. She started studying its toxins, but then got into understanding the regulation, how it alters its lifestyle. And what she's had become a real thought leader in was a process called quorum sensing, where bacteria use small molecules to sense how many others like them are nearby, so that if they have a quorum, enough people, they can switch and produce a particular trait. So she found 2 separate quorum sensing systems in Pseudomonas that equips this opportunistic pathogen to govern when to express its virulence factors and also to coordinate its biofilm development, which Pseudomonas, unfortunately, is a big risk to people with cystic fibrosis because it can use its ability to form biofilms to establish chronic colonization of the lung, which unfortunately are resistant to antimicrobials. So over her long career, she published more than 150 papers and 7 patents, and was just a real thought leader and also an advocate for equal access to our field and also to leadership positions. So I'd love to hear any reflections that any of you have about your time being wowed by Barbara Iglewski or personal interactions you've had with her.
Well, first let me say that I really loved her.
Yeah.
I thought she was terrific. But I was scared of her. She was a scary person. Intimidating about her. And I am intimidated very easily. That kind of thing.
But it didn't get in the way of your friendship, is that right?
Not at all. Not at all.
That's great.
Indeed. I just remember her listening to her when she would come and give her reports about she managed for a long time, I think, ASM publications.
Right, 9 years.
9 years. And I remember listening to her reports on how she was making certain that ASM had the finest books, the finest journals, and she was a good steward of all that happened. I think you probably interacted with a lot, Elio, when you were producing the E. coli and Salmonella tomes that you produced for the society.
Oh, that was, yeah, she did. She played a role, but there are a lot of people there. The main one that worked on that was Fred Neithaler, of course.
Yeah, the former chair of my department.
Yeah.
Of blessed memory.
Yes. Well, we were remembering, Elio, that for episode 200, we came to San Diego and were able to share that with you. I have great memories of that trip.
Had a great time.
Yeah.
Well, it's nice yakking with you.
It's really nice to see you,
Wonderful
Elio.
to see you.
All right, that's TWiM number 300. You can find the show notes at microbe.tv/TWiM. If you like our work, please support us, microbe.tv/contribute. And if you have questions or comments, TWiM at microbe.tv. Our special guest today, Elio Schaechter. Thank you, Elio.
My pleasure, really.
Michele Swanson's at the University of Michigan. Thank you, Michele.
Yeah, go blue. Win that Rose Bowl.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, everyone. And we're still undefeated in football after 200 years.
But yet not in the Rose Bowl, Michael.
And you also have no victories. Yeah.
And we have no victories either. Thank you very much, Michele.
And Petra Levin is at Washington University in St. Louis. Thanks, Petra.
Thank you. It's great to be here, and I'm familially obligated to say go blue.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.